Hello everyone, welcome to the biological database video. If you're a student or a teacher who is completely new to bioinformatics, consider this tutorial as a very basic guide or an explanation to the biological database. Today we are going to look at a biological database which is called as UniProt. UniProt is a part of SIB, that is Swiss Institute of Bioinformatics, and it is a biological database for proteins. Right, so all the information regarding the protein sequences, the protein structures can be obtained from UniProt. So if you go to Google, you type UniProt, and then you go for the first hit. Now, you might have heard of SwissProt also, which is another protein database. So SwissProt and UniProt both are the part of SIB. So even if you type SwissProt, which you can see here, all right, even if you type SwissProt, the hit which is going to be shown to you is about UniProt itself. The reason being is, as you can see on the left hand side, the SwissProt is the manually annotated and the reviewed one, and there is also a tremble which is translated EMBL, that is the translated sequences of EMBL. So both your manually curated, annotated and reviewed data, as well as your translated sequence data, both this data is present in this huge knowledge base which is called as, or a database which is called as your UniProt. Right? So now let's start using this database. So I want to say search a sequence, the different sequences for let's say the P53 protein, right? So I type P53 and then I click on enter. After it happens, then you get a number of hits. So there are two types of hits as it is stated here. You have a reviewed hit, okay, which is manually annotated. This manually annotated data is nothing but extracted, as it's mentioned here, from your literature databases and also from your computational. And also there is an unreviewed data which is from the Tremble, which is a computational analysis. So both this data you will be finding it. So around 92,144 hits we have got for the P53. All right, so here you can see the Tremble data, and there are a lot many reviewed or the curated data. Now if you see on the left hand side, you can see the filters. Okay, there are 2,300 reviewed ones and 89,000 unreviewed ones. Then you have the filters about which organism you want to go ahead with, or then you also have the search results about regarding the gene name, ontology, organism. You can also view them according to the taxonomy, the keywords, etc. So if you see here, if you look at the hits, the first ever hit is about P53 underscore human, because the organism here is about Homo sapiens. The sequence length is 393. So you have for mouse, you have for rat, okay, you have for monkey. So depending on whatever your sequence of interest is, which you are actually finding, if you want to find for a mice, you can get P53 sequence for mouse here. If you want to find for rat, so this is completely dependent upon what you are actually searching for. But let's say for simplicity we are searching for P53 for Homo sapiens. Okay, so then we click on this entry ID here, which is P04637. You can call this as an entry ID, you can call this as an accession number for UniProt. It's your choice. All right, so let's click on P04637. Yeah, all right, so here is the information about your UniProt. So now let's explore the UniProt in detail. Okay, so the first one here is your name, which is your P53. Then you have the gene. So your P53 cellular tumor antigen belongs to the gene name, which is TP53. The organism is Homo sapiens, and it is the status is reviewed. All right, as it's stated here. Now let's explore it, this entire P53 on using this display the left filters, okay, the left column which you can see. You have entry, your function and everything. So let's go for the function here. Okay, we can see it here, but just I'll use this method, okay, for simplicity. So we have the function. So this gives you the function of P53. If you click on the drop down, you can see the 13 publications. You can read all the 30 publications regarding the P53, which is a part of your UniProt. All right. All right, so this is in short the molecular function biological process of your P53. All right, this here gives you the various databases with which your P53 is actually connected with. All right, so this is the Reactome database. Okay, this is Reactome is nothing but your pathway database. All right, the enzyme or the pathway databases. So these are all the list of the pathways. All right, with where your P53 can be involved. All right, the same thing, your protein and group databases. I'm not going into the individual clicking of because then it would take me to a different database and a different explanation overall. I'll again scroll up because as I told you, I will use this left bar here. Next, let's go for the names and taxonomy. So if I click on names and taxonomy, it's going to give me 1 minute. Yeah, if I click on names and taxonomy, it's going to give me the name of the protein, the gene name, the taxonomic identifiers. What is your taxonomy here? The lineages. Okay, this is your taxonomy lineage for our organism, which is your Homo sapien. Next, let's go to, if you click on subcellular location, it will take you here. So subcellular location here gives you all the various places, okay, in your so in your cell where all the P53 acts and it's present. Okay, so all right. Next, let's go to pathology and biotech. So pathology and biotech tells us how P53, what is the importance or significance of TP53 or cellular TP53 in your diseases. We know about cancer very widely. We know it that P53 plays I mean, the defect in P53 or mutation in P53 leads to cancer, as it is said here. Okay, it is frequently mutated and activated about 60% of the cancer. So, which all cancer? What are the different types of cancers? Okay, is what you can see here. All right, so there's a huge list of diseases what you will find when it comes to involvement of TP53 in your diseases. So one thing remember in bioinformatics, your objective should be clear. So what you are searching for, what is the objective for what you are searching? If that is clear, then this searching, okay, of a specific organism, of a specific protein, of a specific DNA or specific gene makes makes more sense. Okay, so this is very important tip which I'm telling you. Your objective should be very clear of what you want to find. So the next is your PTM and processing. PTM stands for your post-translational modification. Okay, and this entire section is about your post-translational modification. So how what are the various genes or the phosphorylation or ubiquitination of what all genes leads how and how does it affect your P53 is what is being explained in your post-translational modification section. Okay. Next, we'll go to expression. The expression here stands for your protein expression. So in your tissue, in your gene expression, how the P53 actually plays a critical role. If you go to any of these gene expression databases, you'll get to know more in detail about it. All right. Again, you have the gene ontology molecular functions, and then finally you have your protein-protein interaction databases. What now? Okay. So the next one here is your structure. If you see the structure, this gives you the information about all the protein structures or the PDB structures. Okay. If you see this one AIU one, sorry. structure, I'll click. Yeah. So if you click on one AIU, one AIE, one C26, so this is one AIU. If I click on one AIE, the structure would change. Yeah. So these are all the structures which are associated with your P53. And if you click on any of these links, it's going to take you to, so this is an NMR structure of which is 1DT7, which your position of 367 to 388. All right, so this is one. So if you click on any of these links, it will take you to this respective structure which is there. So there's a huge list of structures, all right, of uh, which are associated with your P50. All right. um, okay. The next one is the sequences. So this is your sequence for which is here. This is your sequence for P53. You can click here and download the faster sequence, all right, of the P53. Next is your cross references. So there, if you want to go through various web resources of whatever you have seen on the top, you can see. You can go above, you can look at the various sequence databases of your choice, 3D structured databases. So this is the entire information of your cross referencing. Then you have the entry information. So your this sequence was actually entered in, I think, 2009, and there's a last modification in 2020. This goes to show that every time there is something new, this P53 human gets mutated. So that's one good thing. So if you can see here, this is the version 287, all right, of the entry, and the version 4 of the sequence. So the sequence has been modified 4 times, and this entire information has been modified 287 times. So we need to be rest assured that whatever information we are getting is correct, because the last modification or the last update for this P53 here was August 12th. So past, that is, yeah, from 2009 to 2020, every time in 11 years, a lot of modifications which has been done, so we don't need to worry about. And last is your miscellaneous section. All right, so this is I'll go directly to entry. This is how to actually look, or to this in detail study of your UniProt P53. I hope you have liked this video. Uh, Please subscribe to the channel and do not forget to like it. Thank you.